everybody. Thanks so much for having me today. It was a true treat to be invited to speak here. Apologies, my title slide is a little different from your program, but I promise the content will be on in alignment with what you guys were expecting, hopefully. So what we're going to talk about a bit today is essentially clinical trial readiness, and you've heard a lot about different medications that are on the horizon, different treatments that are on the horizon, and I'm hoping to give you guys a bit of a primer on some of the genetic targeted therapies, which is one of the areas of expertise in our center at UT Southwestern. I'm not specifically involved in any of the development programs for Dravet syndrome, so I'll talk a little bit more broadly and then give you a couple of snippets that the foundation helped me pull together. So here are my disclosures. So I don't think this audience necessarily needs this slide, but I feel like you can't start a talk at a Dravet conference without at least acknowledging what the syndrome is. But you guys live this every day, and you are certainly the experts in this disorder. But I did want to give a little bit of a genetic primer because we're going to be talking about some genetic disorders, or genetic treatments rather, and throwing some terminology around can get confusing unless we've at least set the stage of what we're talking about. So on the left-hand side of your screen, you'll see a neuron. It's a brain cell. And these are the cells that are in the brain and responsible for sending signals to talk to each other that make me talk right now and move my arms, and are also responsible for the seizures that you guys see in your children and in our patients. On the right-hand side of the screen, you'll see schematics of DNA, RNA, and proteins. So DNA and RNA are both genetic material. Our DNA, the top strand, is the double-stranded figure, and it is what is storing essentially the giant recipe book that makes us humans. It is stored in this part of the neuron, the nucleus, and it gets transcribed into RNA, which then gets translated into proteins. So essentially, RNA is that intermediate step between the storage or the recipe and then the protein that gets made and is used by the brain to function. So if you think about it, the SCN1A gene is stored in the DNA, gets turned into an RNA that then gets made into the sodium channel. So with that, that's what causes Dravet syndrome. So a single copy of the SCN1A gene having a mistake in it is enough to cause disruption in some cases of that sodium channel function. And depending on how impaired the function is of that sodium channel determines what a patient's symptoms might be. So on the more severe end of the spectrum, we have Dravet syndrome. But on the milder end of the spectrum, we have other conditions. So with that little primer, I want to dive into some targeted therapies and precision therapies. So we're going to highlight a couple of these. The gene therapy and ASOs, or antisense oligonucleotides, are the things that we're going to talk about in more detail, but you just heard a little bit about the ketogenic diet. In some cases, that could be considered a precision therapy. And then in our first talk with zebrafish, we heard about different molecules or medications or drugs, and those could also be considered precision therapies in certain cases. So I like this slide to start out before we dive deeper into the actual treatments and how we test them, because you've gotten to see a bit of all of this already, even just this morning, and I'm sure even more over the last couple of days at the conference. So I live in the world of patients. So this little blue figure, I work mostly with kids who have autism, and so that's where this figure comes from, but it's really widely applicable to different disorders. You could consider this a patient with epilepsy. We define what that patient looks like in clinics like mine and the others that you guys go to, looking at brain imaging, looking at EEGs, looking at blood biomarkers, all kinds of different ways that we can define what a patient with a particular syndrome looks like. And then people will look at different animal models. Apologies, there's not a zebrafish on this slide, but it would fall into this category. And then you can also look at cell lines. Often patient groups will donate skin cell biopsies, and then those can get turned into cell lines and be studied in the lab. And then we have people who can look at different compounds, either making a brand new compound, a new way to treat it, or repurposing a compound or a drug that already exists on the market. We test it in the animals, we test it in the cell lines, and then eventually the goal is to get it back to patients, of course. And so through this process, we can identify underlying pathways, we can identify potential targets for therapy, and maybe even specific populations who may benefit particularly well from a certain treatment. And so then we can think about what are some of those precision therapies, and the two I mentioned I'm going to focus on are the antisense oligonucleotides and then the gene therapy, because these are some of the things that are coming up the pike for you guys too. So remember when we had that figure of DNA. So DNA is where the message is encoded, or the recipe. That gets turned into an mRNA or an RNA molecule that then gets made into a protein. So this central part of the figure is what's naturally happening in the patient who has a mutation in that DNA copy, and they have a protein that is not functioning properly, this purple protein. Antisense oligonucleotides can do one of two jobs. To make it pretty simple, they can either stop manufacturing of that mutant protein, or they can upregulate or increase manufacturing of the healthy copy of the protein. In SCN1A, you've got one bad copy and one good copy. So the goal of the antisense oligonucleotide is to increase the production of the the healthy protein, the healthy copy. So that's what's coming out of Stoke Therapeutics. There are actually trials already underway for this compound. The results from the initial few patients, I understand, I've not been involved in that analysis, but I'm told are showing some promise. And this is targeting kids ages 2 to 18 years old. And this is through repeated intrathecal injections. What does that mean? So we heard just now a lot about lots of different ways to get medicines into the gut or the bloodstream. Intrathecal means that the medicine's going directly into the spinal fluid. In many cases, that's through a lumbar puncture, so a spinal tap, or for moms who had an epidural, kind of a similar procedure where you get a needle in the back and medicine gets injected into the spinal fluid directly. Antisense oligonucleotides have to be redosed every so often, usually on the range of like every 3 to 4 months, but each compound's a little bit different. So I would direct you guys to Stoke or your foundation if you have additional questions about this potential treatment. And then gene therapy is a little bit different. This is actually using DNA. And so the goal of this is to essentially have a Trojan horse that takes in the healthy copy of the human gene. So what we've done, and I say we in the field of gene therapy, I have not been involved in Dravet specifically, but we can take what's called an adeno-associated virus. This is a virus that's naturally occurring in nature, but it doesn't cause any human illness or disease. We can take out the viral genes, so the genome that is natural to the virus, you can actually take out in a lab and then plop in your Dravet gene, for example, and then you deliver that to the patient. It can be delivered in a lot of different routes. You can give it intravenously, so through the IV directly into the bloodstream, but you can also give it directly into the spinal fluid, just like they do with the ASOs, either intrathecal or a number of other routes of administration to get it directly into the brain and spinal fluid. So this is what's coming out of Encoded Therapeutics. Tentatively, they think this trial will begin later this year, and they are targeting younger children in this case, so ages 6 months to 5 years old, and this is going to be a single injection directly into the brain, actually. So really exciting things coming up. So what are some of the risks with gene therapy? Of course, there's a procedural risk. As I mentioned, a lot of times we're going directly into the brain or the spinal fluid around the brain. In this case, this is going to be a direct injection into the brain, and I'm not involved with Encoded Therapeutics, but more broadly speaking about gene therapy, we always think about those risks of any time you are going into the brain space. There's going to be a risk of infection, certainly pain or bleeding. Many times kids will need to be sedated for these, so there can be some procedural risks around that sedation. And then, of course, considerations around medications and what else they're taking. Inflammatory risks are one thing that you'll hear a lot about in the field of gene therapy very broadly. So we are injecting a virus. The virus is going to rev up your immune system. So just like if you get the common cold and you get a big inflammatory response, so maybe you get fever, myalgias, muscle aches and pains and weakness and not feeling good, you can get those same kinds of symptoms after a gene therapy. But you could also potentially have a reaction to the protein that gets made after the gene therapy. So remember, we're delivering a way for the body to now make a healthy copy of that protein. And maybe the body hasn't seen that protein or could see that as a foreign invader and have a reaction to that protein too, in addition to just the reaction to the virus itself. So there's a lot of ways that we try to minimize that and keep patients comfortable, but also may include some immunosuppression, so maybe some steroids or even other medications to kind of decrease the immune's response, the immune system's action against that treatment. The long-term risks are really not known very well yet. We're still in the very early days of gene therapy, and so there's a lot left for us to learn. I think it's a really exciting time, but I think we also need to have our eyes open and be observing and monitoring closely and relying heavily on parents reporting to us. What are the things you see in your child? Are there anything new, anything concerning? And just having lots of open discussions if you are a participant in a clinical trial for a gene therapy. And for gene therapy, it's a single dose injection typically, and we can't really go back and give it again. So I mentioned with the antisense oligonucleotides, that's a repeat injection, a repeat treatment. You're going to get it every few months. But with gene transfer therapy, it's really thought at this current stage of technology to be a one and done treatment. We give it one time, the body mounts a really big immune response to it, and it remembers that. So just like when you get vaccinated or you've had an infection, your body is kind of primed to fight that infection the next time it sees it. It's going to be ready. It's going to stop it. It's similar with the gene therapy. Excuse me. So your body will be primed to fight that infection the next time. Even though the infection is actually a healthy treatment that's going to be good for you, your body sees it as an invader. And so your body will fight that. It will kill those viral particles, even though they have a really good thing for the body instead. So lots of people are doing a lot of work on the immune aspects of gene therapy, but for now, we're in pretty exciting times, and many clinical trials are using similar technology to treat lots of different conditions. So for the rest of the talk, pretty much, we're going to talk more broadly just about clinical trials and how do you actually take things that get discovered in a lab and transfer them into human patients. So our clinical trials are divided into phases, typically phases 1 through 4. You may have heard this if you've heard industry partners or talk about, oh, this is a phase 1 study or this is a phase 3 study. This is what that means. So we always start with preclinical, looking at the mouse models, the zebrafish, cell lines, things like that, and make sure that it's safe and we believe it's going to be effective. We think it's going to help patients. Typically, in most drug trials, you're going to go into a healthy population first who doesn't have any disease of any kind. These are healthy volunteers off the street. They sign up to try a new compound. But in the world of gene therapy, we're often going in to our disease population first because we don't typically want to give a gene therapy product to a healthy volunteer, especially since I mentioned with gene therapy in particular, that's a one-time shot. It's permanently there in your body. We don't want healthy volunteers signing up to get an extra copy of SCN1A, for example. So we're most often having a phase 1-2 study for our initial gene therapy trials. There may be some caveats to that when we get into other types of gene therapies, and there's always going to be exceptions to the rule, but in general, this is where we're headed. The phase 3 study is mostly looking at efficacy, while that earlier phase 1-2 study is really primarily looking at safety. Do patients tolerate this drug? Do patients tolerate the procedure and the treatment and all the other things that go with it? Phase 4 studies come after the drug's been approved, and those are for continued monitoring to make sure that we have a longer time period to see how well is it working and is it still safe and tolerated well. This is the next 2 slides just to show you how the FDA processes these new drugs. It's a super complicated process, and that's the main takeaway, is that you go through the drug development pipeline. It has to have lots of extensive testing on the front end before you ever touch a human patient. You go through a very intense application process called an IND application, or investigational new drug application, and then you walk through all the different phases like we just had in our last schematic, and eventually you get to the market where it's available more broadly to lots of different patients. And so how do you actually conduct a study? So now we know the groundwork of here's what they are, here's all the different phases of the treatment, but how do we actually design the clinical trial? Well, rare disease research is messy, and Dravet syndrome, of course, still counts as a rare disease. It's one of the more common rare diseases, but still rare nonetheless. How do you know that they wouldn't have had this outcome if you hadn't had your special intervention? Maybe it was just that their seizure medication started working all of a sudden. How do you control the environment? You guys are living in lots of different places across the country or the world. You may have different doctors helping you. There may be different norms in different parts of the world. And how do we control all those different variables in patients? In contrast, in the animal models, you control everything that mouse goes through in a day. But in humans, we're a lot messier. And then how do you balance these risks and benefits and timing of an intervention? How do we know when is too early or when is too late? How do we know how long we should watch it? All of these we get clues on based on the animal studies, but there's sometimes still questions, and we make our best educated guess as we're designing the trial. Now, the gold standard is to have a placebo-controlled, double-blinded, randomized clinical trial. You may have heard that at some point. It's a mouthful, but essentially what it means is we take a whole group of patients and we say we're going to randomly pick who goes into the intervention group and who goes into the placebo group, or the dummy group, or the group that gets the sugar pill, for example. So everybody gets split into these two groups. They get treated exactly the same way over the course of the trial. Nobody knows who is in which group, so the patients don't know and the investigators don't know. And then at the end of the trial, you unblind everybody, so you reveal who was in which group, and you look to see, did my treatment group or my experimental group do better than my non-treatment group or my control group? But in rare disease, this can be kind of tricky because we usually have smaller populations, so you usually need a pretty big population to pull from and to enroll in a study like that. And then you can think about, if we're doing an invasive procedure, how do you make that a true sham procedure where patients really don't know if they got a lumbar puncture or not? And so it gets a little bit trickier as we get into some of the gene therapy trials. But we always need to think about who's our control group. How are we going to compare? In the case of the randomized clinical control trial, we have a comparator group built into the protocol. So we know that we're comparing to the group that did not get the treatment. But in some cases, we may not have that control arm. So how do we pick? Either we can control and everybody gets compared to themselves. So what did you look like at the beginning and what do you look like at the end? Or sometimes we'll compare to a historical group. So we know that Dravet patients look a certain way at certain ages over time. And we can say, how does a 3-year-old Dravet patient in this clinical trial look compared to what we thought they might have looked otherwise? So in the fenfluramine trial, this is a great example of a placebo-controlled clinical trial that your population may be kind of familiar with. It was a small compound that was administered like a medication or a drug that you would have at home. They followed patients over 12 weeks who got either the fenfluramine or a placebo or a sugar pill, and then see the difference between them. And they could clearly see in the graph on the right that you've got your fenfluramine patients on the top and your placebo group on the bottom, and the fenfluramine patients had a larger seizure reduction, so had a better response to the treatment. Now, if we don't have that control group, what do we do? So a couple of alternative designs that are coming up very commonly in rare disease, and so I like to give populations an idea of what these are, is N of 1 and placebo phase trials. There's a lot of different ways to skin this cat, but I'm going to just focus on these two just for the sake of time. So an N of 1 clinical trial design just means that we have one participant in the trial. So N is always the number of participants that we have. So N of 1 is one single participant. So what we do is we take the patient or participant, we enroll them in the study, follow them for a preset period of time, do some assessments, maybe that's cognitive or behavioral testing, you complete a seizure diary, EEGs, things like that. And then you give them the treatment, and then you measure your outcomes. So the same things that we use to test at baseline, we're going to test after treatment. And then if you can, maybe you give a washout period. What's a washout period? So that's where we think that the drug has left the system completely, and we think that they've returned back to who they were before. So let's say it's a seizure medication. You give it for a period of time, you stop the medication, and you watch how they do. You give it again, you see how they did, you stop it, and you see how they did. And you do this kind of up and down. Now with gene therapies, again, one shot time, so we don't have the luxury of those later controls and how do they do in the washout period. With antisense oligonucleotides, it's still a little tricky. There might be a way to do it, but it's still pretty hard. But this is what the N of 1 trial design would be. So that patient essentially serves as their own control. How do they look with and without the treatment? The randomized placebo phase trial, though, is one that I think is becoming much more popular in this arena, and especially with gene transfer therapies, because everybody will get treated eventually. It's just that your timing of treatment is the part that gets randomized. So the group of patients and participants is identified at the beginning, and then you randomly get assigned to get treated at a particular time. So there'll be a patient that gets treated first, and then second, and then third. And then people like me and the assessors, so maybe your speech therapist or your physical therapist who's doing some of the assessments, or the psychologist that's doing your cognitive and behavioral testing, they don't know who's been treated and who hasn't. So we don't have them biased, or we don't have them hoping that a patient's doing well and maybe inflating your scores a little bit. And so this adds a little bit of rigor, but everybody still gets treated. You have a smaller pool of patients, but you still have some degree of control within the clinical trial. And then if neither of these are good options, then we need to think about what are our observational studies, how do we know what patients typically look like, and even if we do use one of these trials, these observational studies are super important. And some of you may have participated in some studies like this, where you're not getting a treatment, but physicians like me and investigators are just curious about what do your patients look like, what do your kids look like, how are they doing, how do they do over time. And these can come in a lot of different flavors. So there's patient registry studies where you're maybe filling out a bunch of surveys online, which I know take a lot of time, but they are immensely helpful. Retrospective studies where we can just take all of your medical records, we comb through them, and we do a deep dive, and we try to pull as much really good information out of those medical records as possible. And then prospective studies, or looking forward studies, patients get enrolled in a clinical study, and they get followed at regular intervals, so maybe an EEG gets done every single year at the same time, regardless of how you're doing seizure-wise. Maybe you get an image done at a certain time interval, or they do blood testing, or they do regular psychological testing to see how are their problem-solving skills doing over time. And then also biomarker studies. So do EEGs look a specific way in a particular syndrome? Do we think there's a marker we might be able to follow and have an additional way to assess if our drug is having an effect? And so that's where these historical controls come into play. We really pull them from our observational data and our clinical studies. So a great example that I think has made its rounds is the open label with historical control study that was done for spinal muscular atrophy. So they did a really great natural history study for these kiddos that have a really debilitating neuromuscular condition that causes severe muscle weakness and deterioration very early in life for the type 1 patients. So these kids were either passing away from their disease or ending up on a mechanical ventilator very early in life. And the different colors represent the different ages of the different subtypes. So SMN the number of copies they have of that particular gene. But what they did in the clinical trial for the gene therapy, which was the first gene therapy approved for kids with a neurological condition, Zolgensma is the trade name for that drug, is they plotted out how each child did, and each time you see a tick mark down, that is an individual in the observational study who either hit the endpoint of death or mechanical ventilation. And you can see that there's quite a few in the natural history study who met those endpoints, unfortunately. But in the clinical trial with the gene therapy, that's this blue line across the top, nobody hit those endpoints. And so this was really compelling evidence that we know what patients did without the treatment, and here's what they look like with the treatment, even though it wasn't a controlled study, and we didn't have like a placebo in that clinical trial. Now, death and mechanical ventilation are really dramatic and severe endpoints, but there's other endpoints that we can think of. So we can think about biomarkers, so things like blood testing, maybe your genome or protein levels, EEGs or event-related potentials, ERPs, those can be like visual or auditory evoked potentials, and maybe there's an imaging finding that we can follow on brain MRI or MR spectroscopy. But then there's also clinical endpoints of how do patients feel or function or survive. And so these are things like your psychological testing. So when kids are going for their IQ testing or their behavioral testing, maybe you guys are filling out surveys about your quality of life. How do you think your son or daughter is doing, and how is your family doing? There can be disease severity scales also that you guys can complete or clinicians can complete. And then also lots of different surveys that different providers can complete. So we use all of this information to try to help us determine who's responded to a treatment and who hasn't, as well as looking for, is it safe or not? But being a research participant is really hard. It's hard on your kids, but it's also hard on you guys. There's scheduling. Taking off of work can be a challenge. Getting all of the paperwork in order just to even be able to go and participate. The travel to the site is often a lot of work for you guys. It can also be a big financial burden, both the loss of work as well as the added expense of travel. And then just the stress of it. How do you juggle being in a participant in a trial while also managing your house at home? And how do your other kids do? If you have other kids in the home, they're all affected by it. So just know that investigators think about these things as we're trying to plan the trials, and it's really helpful to get information and input from groups like this that can say, I think that's one too many assessments, or no, we can't do 10 surveys. Maybe we can pick the 5 best that we want to do. But then even when you're there, you're finally, on site and participating, there's going to be a lot of downtime. There's going to be a lot of waiting. If you're in a clinical trial testing a new drug, maybe that's going to add some extra stress. Did you do the right thing? Did you make the right decision? And worrying about the unknowns. And then your child's going through a lot of procedures. They're getting poked and prodded and messed with, and none of that is ever fun. And sometimes these can be days worth of assessments. So I think it's important for both sides of us to acknowledge that these are hard things to do. It's really, really important because it changes the landscape of how we treat patients and what we can do for your generation as well as the next that's coming. But we understand it's really hard on you guys. So one of the silver linings of the pandemic has been televisits. So both clinically, but also they're making their way into research. So we're doing a lot more remote collection where we used to just make patients come to sites and stay for days on end. Maybe we can do some of those assessments by just a Zoom visit. Maybe we can record a video assessment of you helping us be a neurologist in your house and having your child do different activities. And that could suffice for instead of coming all the way onto site to have an assessment done. Maybe we have a video check-in to say, how are your seizures doing? Do you have any side effects? Rather than you coming all the way to clinic to have those things done. And then can you fill out any of your surveys remotely instead of coming online, coming on site to do those too? So I will say people are getting a lot more creative about what parts do we absolutely have to be in person for, and what parts could we do remotely that might be a little easier for everybody. And finally, I always like to end rare disease talks with this, Horton Here's a Who. I think you guys probably know this very well, but having an organization behind you really moves the needle. So you guys get to have a collective voice. You get to have a say in what your foundation is supporting, what type of research are you guys funding and pushing forward, and then also having a voice that regulators like the FDA or the EMA can say, yes, our patient community is behind this treatment. These are the things that are important to us, and this is why we think this is an important project to push forward. So thank you guys for everything that you do. And then I will end there, and I'm happy to take questions.
Questions? Okay, thank you very much.